Hello students, we have started with lesson number 1, that is reproduction in lower and higher plants. In the last lecture, we have started with sexual reproduction. I have introduced you regarding the structure of flower. Structure of flower consists of two different essential walls. The first is called as androecium and second is called as gynoecium. Now in this lecture, we are going to study the structure of anther, which is a part of stamen. In the last lecture only, I have introduced regarding the sexual reproduction, which is divided into three stages: pre-fertilization, fertilization and post-fertilization changes. Pre-fertilization changes consist of maturation of essential floral whorls. Now very important point regarding the generations. Each angiospermic plant shows two generations. One is sporophyte, which is a dominant generation, undergoes meiosis to produce haploid spores. Those haploid spores undergo germination and they produces the gametophytic generation that develops inside the flower only. Gametophytic generation is a haploid one that produces male and female gametes. Now starting with the male reproductive whorl, which is called as androecium, that consists of members. The members are called as stamen. Now here you can see the structure of stamen, which consists of the upper fertile sac-like portion, anther. The stalk is called as filament, and the point where the stalk or filament attaches to the anther lobe is called as connective. Sometimes the anther may be a single lobe, which is called as monothecous. Here the anther is bilobed, consists of two theca, two sac. Internally, the bilobed or dithecous anther shows four microsporangia groups, and that is called as tetrasporangiate anther. In the immature stage, anther consists of a group of parenchyma tissue covered by a layer of single epidermis. Then differentiation takes place in the tissue and hypodermal cells, they are transferred into the inner archisporial cell. Internally, the TS of anther consists of the sporogenous tissue, which is present at the center, covered by the primary parietal cells. When it undergoes development, the sporogenous tissue forms the microspore tetrad, and the primary parietal cell shows or converted into, develop into the four different layers, forms anther wall. Here you can see the TS of tetrasporangiate anther. When it becomes mature, from the outermost it shows the four different layers. The first is called as epidermis, which is a layer of flattened or rectangular cells which are tabular cells. As outermost function is epidermis. Inner to epidermis, it shows a single layer of cells. Here you can see, which is called as endothecium. The cells of endothecium, they are elongated and they show the presence of fibrous thickenings. The function of endothecium is the cells of endothecium, they loses water at the dehiscence. When the anther opens to release the pollen grain, fibrous thickening cells, they undergo opening, helping dehiscence of anther. Inner to endothecium, 1 to 2 layers of middle layer are present. Here you can see. Middle layer, they undergo degeneration because they do not place any function. Then in the center, you can see the four sporogenous cells, sporogenous group of cells. They are covered by a layer tapetum. Tapetum is the innermost nutritive tissue which gives nutrition to the developing male gametes. The sporogenous tissue consists of diploid microspore mother cell undergo meiosis to form spore tetrad or pollen grains, is called as microsporogenesis. And here you can see the vascular trace, which is called as connective. Sporogenous tissue produces the microspores which undergo maturation to form a pollen grain. Now starting with the structure of pollen grain. Here you can see a diagram. Pollen grain, these are the unicellular, uninucleate haploid and non-motile structures. Each pollen grain consists of the outer layer, which is called as sporoderm, inner cytoplasm, and having a nucleus. Sporoderm shows two layers. The outer is called as exine, which is a thin. It consists of the openings, those are called as germ pore. The exine, it is non-biodegradable. Germ pore help to emerge the pollen tube during the germination of pollen grain. Inner layer is called as intine, which covers the cytoplasm, and here haploid nucleus is present. Now next point is development of male gametophyte. The first stage of male gametophyte is the pollen grain. This pollen grain undergoes two successive mitotic division. When it undergoes first mitotic division, the next stage is called as two-cell stage. After first mitotic division of pollen nucleus, it forms two unequal cells. The first is called as vegetative cell, which is irregular in shape, consisting a nucleus rich in cytoplasm. It is rich in the food also. The second, smaller, is called as generative cell. Generative cell attaches on the wall of the vegetative cell, or it is suspended freely in the cytoplasm of vegetative cell. In most of the angiosperm, pollen grains, they releases from the anther in two-cell stage, and they deposits on the stigma for further development. After the two-cell stage, this two-cell pollen grain deposits on the stigma, where the second mitotic division takes place. The generative cell undergo mitosis to form the two haploid male gametes. The structure consisting the pollen tube consisting two male gametes having the tube nucleus at the tip. This structure is called as male gametophyte. Now at the last, the property of pollen grain, which is called as pollen viability. Pollen viability is the ability of pollen grain to germinate on the stigma. It is variable. It is depend on the external climatic conditions like temperature and humidity. In case of monocots like maize, wheat and the rice, the pollen viability is of 30 days. In some angiosperms, which belongs to family Leguminaceae, Solanaceae, Malvaceae, pollen viability may over the 1 month. So this is regarding the structure of male reproductive valve. I am sending some questions depending on this topic. In the next lecture, we will start with female reproductive valve, that is gynosphere. Thank you.